Yes, all the cells of our body need to divide. Okay, of course they need to divide because we are all growing, right? The cells are either growing in size or they are growing in number. Okay, whether they are growing in size or they are growing in number, they need to grow. And since they need to grow, then this means that every cell of your body is in some developmental stage of growing. And that stage or that cycle of growing can be summarized in this pie chart, which you can see right now in front of you, right? It shows you that this is how a cell undergoes a process of cycle all the time in the body. And every cell is in some phase, right? For example, you can see that there is a pink colored phase which has been labeled as, now I'm going to zoom into it, it has been labeled as interphase. It has been labeled as interphase, and it starts from this one, G1 plus S plus G2, right? For example, just look at my cursor.
Your voice is not audible.
Achha. Can you hear me now?
Yes.
Is it clear now?
Yes, miss.
Huh? First, it was not too clear. Just tell me whether it's clear now.
Yes, miss, it's clear.
Okay. Now see. So this is the entire cell cycle. Okay? And the cell cycle, I can say that the longest phase of the cell cycle is known as interphase, okay, which is starting from this arrow and it is going to this arrow. And if I want to actually write down what is interphase made up of, so I would say it is a combination of G1 plus S plus G2. क्या मतलब है इस बात का? It means that a cell when it is growing, it is synthesizing proteins. जैसे कि अगर मैं कहूँ कि you've done biological molecules, isn't it? So if it is a red blood cell, it should have the protein hemoglobin. If it is your tendon, it should have the protein collagen. If it is your muscle, it should have the protein myoglobin. And if it's an enzyme, it should be made up of, like, enzymes, of course, are different kinds of proteins. So basically, almost, and hormones also, and neurotransmitters also. So her work body may different kinds of proteins, they need to be made. So let's assume that there is a tiny cell over here, and like any random cell of your body, it needs to make proteins. And to make proteins, what does it need? It needs the organelles to grow. It needs the protein synthesis to happen. So ये जो पूरा जो आपका phase है ना, ये G1 वाला. Now I am actually highlighting G1. G1 is starting from here and G1 is finishing here. ये मैंने दो इसके वो बना दिए. This is G1. So I would say protein synthesis is taking place in G1. And protein synthesis means different kinds of proteins हमें, इतनी तो हमें proteins चाहिए रोजाना, right? We need hemoglobin, collagen, elastin, myosin, actin, list goes long. We need protease, amylase, lipase, etc. So ये proteins बन रही हैं cell के अंदर. This is also a cell cycle, a part of a cell cycle. अच्छा, now the proteins are made. ठीक है, काम हो रहा है. What if the cell gets a signal that besides making proteins, you also have to increase in number? For example, every day, lots of your skin cells, they die, they exfoliate. You know, exfoliation भी करते हैं कभी कभार जब हम scrubs use करते हैं, and they are replaced. The old skin cells are replaced by new skin cells. The old RBCs are replaced by new RBCs. Similarly, the old cells of the lining of your gut are replaced by new cells. So अब ये जो replacement चाहिए ना, so this is actually, you know, we want now cells to increase in number also because they are dying. Not only the cells are dying, even the organelles are dying. अगर आपको याद हो कि हमने किया था ना कि one of the functions of lysosomes is to digest old organelles, old mitochondria. याद है?
Yes, miss.
Right? So अब cells को number में increase होगी. So this means that let's say if a cell decides, or if a cell, it can't decide, of course, if a cell gets a signal, now comes the concept of cellular signaling or cell signaling, that it has to divide in number. It should, before division, it should undergo DNA replication. Replicate होना चाहिए ना, क्योंकि देखो, for example, you have a cell like this. Okay? This is the parent cell and it has 46 chromosomes, which means 2 sets of 23 chromosomes. You want this cell to divide in such a manner कि आपके पास जो end product हो ना daughter cells का, इसके पास भी 46 हो और इसके पास भी 46 हो. So don't you think that this 46 wala initial cell should undergo replication to make it into 92, so that when this cell divides, we end up with 246 cells? Yes or no?
Yes, miss.
Okay? So which is why DNA replication is needed. So now you should understand that DNA replication and protein synthesis are different parts of the cell cycle. A cell always, a cell is almost always in some or the other phase of the cell cycle, और ज़्यादातर majority cells जो होते हैं ना वो इस वाले phase में होते हैं. Not all cells are undergoing DNA replication. Not all cells are increasing in number. कुछ cells जिनको जरूरत होती है जिनका lifespan बहुत short होता है. For example, the average lifespan of a white blood cell is, I think, only some days to weeks. ठीक है. फिर इसी तरह से average lifespan of a red blood cell is around 120 days. Skin के cells तो रोज exfoliate होते हैं. Gut के cells भी, gut lining cells are also exfoliated every day. So we want them to grow in number every day. So these are those cells which will receive a signal, and then after the signal is received, DNA replicate हो जाएगा. अच्छा, DNA replicate हो गया, and if you remember, or do you recall the name of that enzyme that is responsible for DNA replication? What's that? The main enzyme that adds nucleotides. Yes, Amna, any idea?
It's DNA polymerase.
Polymerase, very good. And if you remember, I told you that this DNA polymerase is also known as a proofreading enzyme, right? And the chance that it is going to copy or add nucleotides in an incorrect manner, which means the chances of error is 1 in 10 raised to the power 8 billion base pairs, right? So इसका मतलब ये हुआ कि there has to be the cell after DNA replication has to go into a phase, it's a small phase, is known as G2, in which errors, if any, they are checked, proofreading is done, so that the cell can now go into mitosis, जिसमें actually one cell will divide into two cells. Although I'm not doing the chapter mitosis with you, but just so that protein synthesis should make sense to you, and you should know what is the logic behind a cell entering into the phase of protein synthesis and entering into DNA replication. I have shown you this pie chart. So I hope that's clear.
Yes, miss.
ठीक है. अब आते हैं इस बात पे प्रोटीन सिंथेसिस. So just a minute, I just want to switch off the fan, it's very cold. Just a minute. Basically, this process, protein synthesis, is sometimes also known as the central dogma of biology, or the central idea of biology, the entire subject biology, because if you don't know this, you don't know the biology of your body, okay, or biology of any living organism. Every living organism, starting from a tiny prokaryote to a fungus to a plant, a unicellular plant or a multicellular plant, or humans or animals, all of us, we share one amazing molecule, and that's DNA. Okay? So if we all have DNA, this means we all have a set of coded instructions which need to be transcribed into RNA, and the RNA needs to be translated into a protein so that the body can finally know what protein it has to make. Body को insulin बनाना है, right? Let's say कि insulin is made up of these amino acids. ये insulin का formula है. Amino acid A joined to amino acid B, joined to amino acid C, joined to amino acid D, and amino acid E. ठीक है? I'm just assuming, just giving you a hypothetical example. Let's say ये बनाना है. ठीक है? Now this body, or our body, cannot just join up these amino acids through peptide bonds and make an insulin molecule unless and until the instructions, the coded program inside DNA is transcribed into an mRNA molecule, and still the body is not going to understand this language. Nahi samajh aayi, language is further translated into this, what? Into an amino acid sequence ताकि protein बन जाए. So we would say in very, very simple words कि हम स्टूडेंट से पूछते हैं कि बेटा व्हाट डू यू अंडरस्टैंड बाय और व्हाट डू यू मीन बाय प्रोटीन सिंथेसिस, तो ऑल ऑफ देम से टुगेदर दैट प्रोटीन सिंथेसिस इज इक्वल टू ट्रांसक्रिप्शन प्लस ट्रांसलेशन। ठीक है, तो यही आपको आना चाहिए कि प्रोटीन सिंथेसिस मीन्स ट्रांसक्रिप्शन एंड ट्रांसलेशन। Was this much understood from the recording?
Yes, miss.
Acha. When we move forward, I'll just quickly move forward so as to cater your queries also. Yeah, this is very important. Protein synthesis is a two-step process summarized in this diagram. Always remember that transcription happens inside the nucleus and translation happens inside the cytoplasm. ठीक है, ये बात तो बिल्कुल clear है।
Yes, miss.
ठीक है, अच्छा तो अच्छा, now moving on to this, see a DNA strand ATGC and I want to make an mRNA strand from DNA. So of course मुझे DNA से RNA बनाना है, so then in any case just like DNA replication I will have to open this strand, खोलना पड़ेगा इसको through the same enzyme helicase and use one of these strands as a template to make a single stranded mRNA molecule. By the way, M stands for messenger RNA and we always write this word M in small, ऐसे करके लिखते हैं. There's a way to write it. You cannot write it like this. ठीक है, ये कुछ examiner कुछ और ही समझ बैठेगा इसको. ठीक है, and if you want to write DNA is of course in all caps lock. So mRNA or messenger RNA, or you can see that from A, T, G and C, we can make a single stranded molecule which will not contain T, and instead of T, it will contain U. Now let me do a mental exercise with you, okay? I think, yahan nahi hai. Okay, I'll ask you this question. Okay, let me just hypothetically, okay, A, T, G, C, A, T, G, C, A and T. If I want to make an mRNA from this one, ठीक है, इस वाले से, make a complementary mRNA and tell me the basis. For A, what should I write? Yeah, what should I write over here for A? A will pair with what on mRNA? Yes, beta.
T.
But mRNA doesn't have thymine. उसके पास तो thymine है ही नहीं. Thymine के बजाय क्या है उसके पास?
U.
U. So A will pair with U. ठीक है. What is the complementary base for T? T.
A.
A. Very good. What is the complementary base for G?
C.
And for C?
G.
And for again A?
U.
U. And T, let's complete this. T.
A.
A.
A. C.
Huh.
G. And U again.
G. Sorry. G and U again. ठीक है? So the examiner is going to give you a DNA code, and he is going to ask you to transcribe this DNA code into an mRNA codon. ठीक है, एक तो ये इसमें ना इस चैप्टर में there are too many technical terms, and at the same time every technical term has its own specificity. You can't just use whatever technical terms you want to. अगर आप DNA की बात कर रहे हैं, then you will say the DNA code, CODE code. Okay, let me just clear these things to you. So for DNA, we use the word code. ठीक है, DNA is a code. ठीक है, and for mRNA we use the word codons. And codon हम कहते हैं किस चीज को codon कहते हैं? A triplet of 3 bases is known as a codon. 3 bases is called a codon. So this means that, let me make a DNA molecule. Let's say this is DNA, ठीक है? इसमें हम कितने 1 2 3 4 5 6 बना लेते हैं, and then DNA and then RNA. 1 2 3 4 5 and 6. ठीक है, again if I have A T C G A T, So for this, quickly tell me beta, what will be the complementary basis for mRNA जो मैंने ऊपर बनाया है?
Miss UAGC
U and then UA
A
G
GC C
C.
UA
No, not C. Sorry,
UA
C. A के लिए U होगा ना? हाँ, UA. एक मिनट, U and A. ठीक है? So this triplet, and this is also a triplet. Every triplet of bases is known as a codon, but the word codon is only used for mRNA. Only used for mRNA. और DNA के लिए आपको कोड होगा, right? So the DNA code, for the word DNA you'll use code, and for mRNA you'll use codons. ठीक है? Moving on very quickly to again a summary. आपको बता रहा है कि transcription हो रही है and it is taking place in the nucleus. और कैसे देखो DNA is being opened up, और open होने के बाद we are using only, इसको color कर लेती हूँ मैं strand को, this strand, this one. इसको हमने as a template use किया है, and this is the mRNA that we have created. This is the mRNA. Always remember the DNA is single stranded and the mRNA is the DNA is double stranded and the mRNA is single stranded. अच्छा, एक और बात बताओ. When we were doing DNA replication, we needed the enzyme DNA polymerase. When we are doing transcription, which enzyme do we need?
RNA
वेरी गुड, आरएनए पॉलीमरेज. अच्छा, जब हम डीएनए रेप्लिकेशन कर रहे थे तो न्यूक्लियस में बहुत सारे फ्री एक्टिवेटेड डीएनए न्यूक्लियोटाइड्स इधर उधर घूम रहे थे, राइट? ओवर हियर व्हिच काइंड ऑफ न्यूक्लियोटाइड्स विल बी फाउंड freely floating in the nucleus? DNA nucleotides or RNA nucleotides?
It's RNA.
Why RNA nucleotides?
Just because it's the RNA.
Exactly, because we want to make RNA. अगर हमें RNA बनाना है तो हमें RNA nucleotides चाहिए. अगर हमें DNA बनाना है तो हमें DNA nucleotides चाहिए. Okay, so this mRNA is made. Let me just color it red. And once it is made, if you remember when we were doing chapter cells, उस वक्त जब हम nuclear membrane पढ़ रहे थे, I hope you remember that. So structures, there are some structures which can leave the nucleus and some structures which can enter the nucleus. So one of the structures that can leave the nucleus is mRNA. और वो आराम से nuclear pore के through बाहर जा रहा है. अच्छा, अब देखो ये क्या कर रहा है बाहर. You can see that as soon as the mRNA leaves the nucleus, it is going to dock on the ribosome. Dock on the ribosome. ठीक है? और ये रहा आपका ribosome. I'm just outlining it. The small unit of the ribosome and the large unit of the ribosome. और ये आपका mRNA जो है आके dock करता है large unit पे, आके बैठ गया. ठीक है? It's docking on it. And we can see that the mRNA molecule has all those bases जो इसके अपने वो वाले bases हैं, A, U, G और C. ठीक है? And they exist in triplets which are known as codons. ठीक है, अभी भी आपकी बॉडी को ये लैंग्वेज नहीं समझ आ रही है. It needs to be translated. So in the cytoplasm we have some protein making machinery. ठीक है, उस मशीनरी का क्या नाम है? Number one, the part of the machinery, the most essential part of the machinery is the ribosome itself, the small and the large unit. Second part of the machinery is the tRNA molecules. अच्छा, what are tRNA molecules? इसको देखते हैं गौर से. tRNA भी बिल्कुल mRNA की तरह ही एक RNA ही molecule है, but it is different in shape and of course it's different in function. mRNA function क्या था कि it's a transcript that is formed by transcription from the DNA, which will be sent outside the nucleus so that it can be translated into protein. But what is tRNA? T stands for the word transfer. ठीक है, T का मतलब transfer RNA को हम कहते हैं. It has some similarities also and some differences also from mRNA. सबसे पहले similarity कर लेते हैं कि just like mRNA, it is also an RNA molecule, ribonucleic acid. ठीक है, just like mRNA, it is single stranded. दिखने में double stranded लगता है लेकिन folded है. Just see how it's folded. ऐसे करके, and you know, look how it's folded. ऐसे 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 करके and ऐसे. लेकिन है ये single stranded. ठीक है. One end of the tRNA molecule has an amino acid attached to it, this end, and this end has again a triplet of bases attached to it. लेकिन ये जो triplet of bases है ना, ये codons नहीं है. These are anticodons. So इनके तो नाम से ही पता चल रहा है कि these anticodons will most probably as you can see they are going to complementary base pair with the codons. ठीक है, ये complementary इनकी base pairing होगी. और reason क्या है complementary base pairing का, कि हमारे पास जो tRNA molecules है ना inside our cytoplasm, numbers में तो बहुत ज्यादा हैं, but variety में, types में we've got 20. Why? Because our body can make 20 different kinds of amino acids. 20. So since the different types of amino acids are 20, so the tRNAs will be also 20, क्योंकि हर tRNA अपने साथ अपने एक एंड पे, इस एंड पे एक अमाइनो एसिड उठा के लाता है, and it is specific for that amino acid. For example, this tRNA molecule, let me show another diagram which is much better. Am I going too fast, Amna?
No, miss.
नहीं, इसको I'll just show you this tRNA molecule in better picture. Top end, an amino acid at the top, or you can say the site of attachment of the amino acid at the top. Anticodon, which is a triplet of 3 bases below. ठीक, और उसके बाद अगर देखा जाए तो it is single stranded. Look at it, single stranded. ठीक है, it has these hydrogen bonds also and it has anticodon also. ठीक है, अच्छा. Each, look here, the function of tRNA is to carry amino acids to ribosomes. It is single stranded with areas of base pairing. There are at least 20 different types of tRNAs. Each tRNA molecule has a triplet of unpaired bases. ये unpaired हैं, जो pair होंगे codon के साथ. So it has a triplet of unpaired bases on one end, known as the anticodon, and it has an amino acid attachment site for a specific amino acid at the top, के दूसरे end पे. ठीक है? Look at this ribosome, small unit and large unit. ठीक है? What is the biological significance of a ribosome? कि it is the place, और हमने किया था ना chapter cell में कि what is the function of ribosomes? क्या पढ़ा था हमने cells में?
So it helps make DNA.
नहीं, what was one single function of
Oh,
ribosome?
miss. Protein synthesis.
हाँ, अब समझ आया. Now this is, this should make
Yes,
sense
miss.
now. हाँ, तो protein synthesis
Yes,
ribosome
miss.
पे ही हो रही है, right? So here you can see that your textbook says, ये सारे point आपके textbook से ही लिए हैं कि what is ribosome? It's a tiny organ. इतना छोटा कि नजर भी नहीं आता ना. We can't even see it under the light microscope. It is 20 nanometers in size, right? So it's an organelle where translation takes place. They are found in the cytoplasm on the rough ER, okay, made of ribosomal RNA and protein, contains a large and small subunit. If you want to know the composition of this ribosome, so it is made up of ribosomal RNA, rRNA, and it is made up of protein. So what does this actually mean? It means कि अगर हम ये देखना चाहें ना कि how many kinds of RNA we have in our body, so we have 3 kinds of RNA. ठीक है, and all these 3 kinds are very very important. एक similarity तो तीनों में ये है कि they are single stranded. So we have the mRNA, बिल्कुल अलग चीज है. We have tRNA, ये भी अलग चीज है. And then we have rRNA, and rRNA is nothing but ribosomes. और एग्जाम में स्पेशली MCQs में जरूर पूछा जाता है कि what is the ribosome made up of? Sorry, we have rRNA. So rRNA makes up the ribosomes. ठीक है, together with some proteins. अच्छा, let's move on. ठीक है, ये भी हमने कर लिया था. Okay, tRNA molecule coming, carrying an amino acid at the top and an anticodon below, so that it will come eventually, and these bases will come close to these bases. They will always be complementary to the triplet of codons. एक आएगा पहले tRNA, and then what will happen that subsequent tRNA molecules will also come. इसमें देखो, एक तो आ गया amino acid at the top, MET stands for methionine. ये जो MET MET है, it stands for methionine. Let me write the name of methionine. It's an amino acid. Okay. अच्छा. दूसरा आ रहा है. It's also carrying an amino acid. You're not supposed to remember their names, but I'm just trying to, describe them. So leucine. ठीक है. Methionine is carrying the anticodon UAC. Leucine is carrying the anticodon AAU. अब जब ये आके लगता है ना, for example, ये आके इसके बराबर में लग गया. So let me show you the third picture के इसके लगने के बाद होगा क्या? This is what's going to happen. When two tRNA molecules, they come and they lie next to each other, that's the stage where a peptide bond formation takes place between the amino acid 1 and the amino acid 2. Peptide bond ban jayega. And what are we actually doing? We are growing a polypeptide chain from different amino acids. So abhi hamari chain sirf 2 amino acids long hai. So technically we've made a dipeptide, but we intend to make a long polypeptide. You know what? Hemoglobin जब हम आपको पढ़ाते हैं ना biological molecules में, we tell you it's made up of 2 alpha chains, 2 beta chains, ठीक है? Hemo and globin, हम आपको कहते हैं ना heme plus globin से ही बना है. तो इसमें जो इसका globin वाला part है ना, that's the protein. And the globin is made up of 2 alpha and 2 beta chain, और इसीलिए हम इसको कहते हैं कि क्योंकि 4 polypeptide chains हैं तो ये quaternary protein है. Do you know that each alpha contains 141 amino acids? So 141 amino acid एक alpha chain के हुए, 141 दूसरी के हुए. फिर each beta chain consists of 146, another beta chain consists of 146. Just imagine the complexity of this protein. 141 plus 141 plus 146 plus 146. तो सोचो कि ये कितनी देर तक इसकी ट्रांसलेशन चलती रहती होगी, right? अच्छा, ये बन गए आपके. Let's look at the third picture. In the third picture is showing you that the first tRNA molecule that had initially come and docked on the ribosome is now being released. Why? Because it's no longer needed. And why? Because we've already made a dipeptide, which is methionine bound by a peptide bond to leucine. फिर जब उसके आगे वाले स्टेज में जब ल्यूसीन पेप्टाइड बॉन्ड बनाएगा ग्लाइसिन के साथ, तो फिर ये वाला टीआरएनए भी रिलीज हो जाएगा, एंड दिस विल आल्सो बी रिलीज एंड सो ऑन. एम आई मेकिंग सेंस, आमना?
Yes, miss.
ठीक है. अच्छा, व्हाट वाज योर क्वेरी? टेल मी.
Miss, you cleared it very early on, like what attaches to you.
Okay, fine. I cleared it, so then I think I should just. Okay. In that recording, was there any mention of the real world scenario?
No, miss.
No? Achha, so then I am putting this slide in front of you, and I want you to read it so that we can discuss it. When we teach students about protein synthesis, we are always talking about just one ribosome, but in reality, it can't be one ribosome. Okay? Our body is not that simple. If we just think that it's one ribosome, then this means that we haven't understood the body really very well. It's too complicated. इतने लंबे proteins बन रहे हैं. सिर्फ hemoglobin को देख लो. आपकी body में तो रोज ही कितना hemoglobin बनता होगा, right? So in the real world scenario, we have lots of ribosomes on one mRNA at the same time, known as polyribosomes. Look here. Let's say that this is one mRNA molecule. Okay? So you don't have only one ribosome, you have this ribosome, this ribosome, this and this and this. So this means that each ribosome will produce one polypeptide, but different ribosomes will produce different polypeptides. So this will increase the speed and the efficiency of translation. Is that making sense?
Yes, miss.
ठीक है, तो ऐसे ही होगा रियल वर्ल्ड सिनेरियो में. अच्छा, हम बात करते हैं ऑलवेज कि वी टॉक अबाउट कि भाई ट्रांसक्रिप्शन हैपेंस इन द न्यूक्लियस, बट सिंस प्रोकैरियोट्स के पास तो न्यूक्लियस ही नहीं है, तो वहां क्या होगा? अच्छा, प्रोकैरियोटिक प्रोटीन सिंथेसिस इज नॉट अ पार्ट ऑफ द सिलेबस, बट generally you should know that although there is no nucleus in the prokaryotes, but transcription still takes place through some mechanism which we are not interested right now. But translation begins before transcription is even complete. So transcription शुरू होती है और उसकी completion से पहले ही translation शुरू हो जाती है. And ribosomes, we know that prokaryotes have 70S ribosomes, so they start translating as soon as some mRNA is made. So that's about it. We don't know, we don't want to go into the details of that. So, now coming to this part. This has been added to your syllabus this year only. Okay, again, I would like you to put your effort. I'll be waiting. I have tried to make this text, which I have shared in front of you, easy by drawing a diagram on the right-hand side. Use your own brain and tell me what have you understood. I'm waiting. So basically, this topic is that of modification of the mRNA. So this means it actually shows us that when transcription is complete, and even before the cell has to enter into translation, the mRNA transcript that has been made needs some modification, right? It is changed. So this means that actually, let's suppose, let's just assume कि आपने अगर कोई 10 मीटर लंबा mRNA बनाया है, तो actually जो असली वाला mRNA होगा ना, it will be shorter than the original mRNA that was made. Now there are reasons for all this happening. There are many parts on your mRNA which do not code for anything, right? Or maybe they might be playing some role which we haven't understood. Maybe research will prove, but for the time being, they don't code for anything. So the body tries to get rid of those uncoded bases. And those uncoded bases or bases that don't code for any protein are known as introns. And there are various enzymes which will get rid of these structures, just like snipping tools. Those enzymes work like snipping tools and they will snip these introns away. So if the introns are being snipped away, let's say कि अगर यहाँ पे मैंने एक snip किया, I've removed another intron over here. So if I made two cuts, one cut over here and one cut over here, so I'm left with how many fragments? One fragment, second fragment, and third fragment, right? So I end up with these 3 exons, and of course, then these exons will be then utilized accordingly. How? Let me just read from here. It says, I'll read it once again, that in eukaryotes, the mRNA is modified, partly changed before it leaves the nucleus. The original molecule before it is modified is known as a primary transcript. So आपको जो लंबा वाला molecule है ना mRNA का जो originally बनता है, that's the primary transcript. Now this is your new syllabus, and your new syllabus wants you to know the terminology of primary transcript. So अगर आपसे पूछा जाए कि what's the primary transcript, so you will say the original mRNA molecule is known as the primary transcript. What is the name given to the process of modification? The name that is given to the process of modification is RNA processing. ठीक है, तो इसमें I'm just putting a star on these keywords. You need to know them. One step of this processing is known as RNA splicing. So we are splicing it, we are removing sections, we're actually snipping up the unwanted RNA. Okay? The sections which we snip or we removed are known as the introns. Okay? The nucleotide sequences that remain after the introns are removed are known as exons, and they have to be joined together after the removal of introns. ठीक है? अभी introns के उस पे जो है ना काफी debate चल रही है कि what is the function of introns? Do they actually have a role or they don't have a role? We're not so clear about it. Scientists know that the introns in the DNA can help to regulate the activity of genes. देखो कोई चीज भी अल्लाह ताला ने बेमकसद तो नहीं बनाई. So maybe they don't code for proteins, but they might have a role in regulating the activity of the genes. They also know that in some cases, a given primary transcript molecule can be spliced in different ways, alternative splicing. This results in different mRNAs being made from the original primary transcript. And these different mRNAs will produce different proteins when translated. It means that one gene can code for several different proteins or different forms of the same protein. So this is the beauty of genetics actually. कि gene आपके पास एक ही है, right? We say gene is a section of a DNA that codes for a polypeptide. तो एक ही gene different तरह के proteins बना सकती है और different forms of the same protein बना सकती है. How? Through the process of modification of mRNA. So this is like the beauty of genetics. Is that clear?
Miss, a person like a person, gradually change over time, like their looks or features change.
Mm-hmm.
For example, once you approach old age. So, Miss, is it because some of the bases are being altered?
In a way, you are right. And you actually, most of the questions that you're asking me are from the chapter mitotic cycle. In our chromosomes, if you're interested, then I will, cater your query right now only. Okay, look, this is the chromosome, right? The chromosome है ना? So all of us have some bases which are found over here. Let me just color this part. ये जो part नहीं है ये वाला, orange वाला, this, this, and this. These parts are known as telomeres. They are, the analogy of telomeres is made with the tip of the shoelaces. Shoelace की tip पता है ना क्या होती है? अगर वो tip टूट जाए तो क्या होगा shoelace को?
The break open.
break हो जाएगा. So in the same way, we are also having these tips of DNA bases at the ends of the chromosome. For example, if I say this is one chromosome, two chromatids, है ना? One chromatid, ये एक chromatid है, ये एक chromatid है. This chromatid is made up of one DNA molecule and this chromatid is made up of another DNA molecule. और इसके tip पे ये telomeres हैं. Telomeres क्या हैं? These are just extra bases. ठीक है? As we grow, just now you said, as we grow, हम सब जब grow होते जाते हैं ना, और हमारी body में millions of times these cell cycle happen. So with every cell cycle and with every replication cycle, especially, we get rid of some of the bases found here. So you can see over here, ये इस diagram में आपको क्या दिखा रहे हैं? As the cell divide over time, so let's say this is first cycle, second cycle, third cycle, fourth cycle. Do you see that the telomere length is shortening?
Yes, Miss.
Shorten हो रही है ना? So this shortening is actually seen as we age. So of course we change, our features also change, and the reason that a person dies, the reason that a person ages, develops wrinkles on the face, the hair, gets gray and then white, or फिर diseases होनी शुरू होती हैं जैसे high blood pressure, diabetes, cholesterol, it's all because of loss of telomeres. Even अगर किसी को कोई disease हो भी नहीं ना, so nobody is immortal in this world, right? So the reason that all of us are mortal is that as we are growing and as we are aging, the telomeres are shortening. And telomere shortening का मतलब ये कि with every cell cycle we are losing some of the DNA bases. I hope that makes sense.
Yes, Miss.
ठीक है? अच्छा. So let's go back to the protein synthesis. हाँ, this was, ये जो अभी आप हमने किया ना, this is modification of mRNA. अब एक दूसरी modification आती है. Let me give it a heading. And this modification is known as modification of the protein. ठीक है? Modification of the protein. We make a heading. वो क्या हो रहा था? Modification of mRNA, and this is modification of protein. In other words, I can call this post transcriptional modification, and this would be post translational modification. क्या मतलब? Once the protein is made, don't you think we need to add some spices to the protein? हर protein एक तरह की थोड़ी होती है. For example, I want to add a polysaccharide to a protein. I want to make glycoprotein. तो मुझे उसमें एक polysaccharide unit डालना पड़ेगा. Don't we have lots of glycoproteins in the cell membrane? है ना? फिर मुझे जो है ये protein था, it was just a straight chain. It just had a primary structure. I wanted to fold through the acquisition of lots of hydrogen bonds. I wanted to coil into a compact shape to change it into a tertiary structure. मुझे एक और दूसरी polypeptide chain भी add करनी है to make it into a quaternary protein. So this means I want to modify this protein into a secondary or a tertiary or a quaternary structure. Maybe I want to add a prosthetic group. I want to add heme to it. Protein तो मैंने बना लिया globin, but I want to add heme to it. That's the addition of a prosthetic group. ठीक है? Maybe मैंने protein बना लिया, but I think that I want to remove an amino acid, or maybe I want to add an amino acid. एक amino acid निकाल दिया, एक remove add कर दिया, so as to customize and get the protein that I actually need in my body. So all these mechanisms जो आपको नजर आ रहे हैं ना example, they come under the topic of post translational modification, and the examiner will ask you what are the various ways, or explain the various ways by means of which your body undergoes post-translational modification. Is that clear?
Yes, miss.
ठीक है, अच्छा, हाँ, ये वही एक ही चीज है, पोस्ट ट्रांसलेशनल मॉडिफिकेशन. I just want to tell you something, that there are some fixed rules for transcription also. What are those fixed rules? Those fixed rules are, अभी सिर्फ एक मिनट रहता है तो I'll just finish this. The fixed rules are that whenever the translation begins, okay, whenever the translation begins, it's always the tRNA that carries the amino acid methionine as the first tRNA. यानी आपकी बॉडी में जब प्रोटीन सिंथेसिस होगी और जब ट्रांसलेशन बिगिन होगी, so the first tRNA will always be having methionine at the top. And another fixed rule, the first anticodon will always be UAC and the first codon will always be AUG. अब इसको याद करने का तरीका क्या है? What I tell my students is schools open in August. AUG August. अगर August याद हो गया तो इसका कॉम्प्लीमेंट्री आप बनाओ, क्योंकि ये भी RNA है और ये भी RNA, तो आप कॉम्प्लीमेंट्री बनाओगे. A के साथ आप बनाओगे U, अगेन इस U के साथ बनाओगे A, and G के साथ बनाओगे C. So if you remember AUG, it's very easy to remember UAC. So the first codon in translation is always AUG. Why? Because schools start in August. Schools open in August, and the first amino acid is always methionine. उसके बाद कौन सा amino acid है, that depends upon what is the requirement of the body. Is that clear?
Oh yes, miss.
Okay.
Is the DNA, sorry, protein synthesis complete at this, like once the tRNA sticks itself to the mRNA?
Yeah. It's complete. It's complete. In fact, I think what was left was, ये चीज कि when two amino acids are brought close together, there is a peptide bond formation between the two adjacent amino acids, and the enzyme that is needed for this peptide bond formation is known as peptidyl transferase, and this process requires ATP. So this means that DNA replication also required those activated nucleotides that have two extra phosphates, और जब वो phosphates release होते थे तो फिर energy requirement उसके लिए होती थी, चाहिए होती थी in the form of ATP. So DNA replication is also an active process, and transcription and translation are also active processes, क्योंकि transcription में भी आपको ATP चाहिए, इसमें भी आपको ATP चाहिए. Why? To make the peptide bond. And in order to make this peptide bond formation, we require the activity of this enzyme, and this enzyme requires ATP. This is the thing which I did not teach. ठीक है?
Yes, miss. This protein synthesis only produces proteins and not DNA, right?
Not at all. Protein synthesis का मतलब ही है produces protein.
Some proteins are made of RNA.
Proteins are made up of not RNA. Proteins are made up of what? You've done biological molecules. Look at this. ये खत्म हो गई protein synthesis. Okay.
Amino acids.
No. Amino acids. Proteins are made up of amino acids. देखो, इधर देखो एक second. Which diagram should I show you? हाँ, look at this. ठीक है? Look at this. इसको देखो आप. First tRNA इधर चला गया, UAC वाला, AUG वाला, क्योंकि हमने इसको यूज कर लिया. And with that first tRNA we made methionine. ठीक है? फिर second आ गया, वो क्या लेके आ रहा था अपने साथ? Leucine. Methionine and leucine have a peptide bond in between each other. ठीक है? अच्छा, अब जब leucine glycine के साथ peptide bond बनाएगा तो फिर ये भी यहाँ पे discharge हो जाएगा. See what's happening? This will also go. And as translation is progressing, our polypeptide chain is growing longer and longer. नजर आ रही है आपको? Growing polypeptide chain. So what are we ending up in protein synthesis with? We are ending up with the manufacture of a polypeptide chain which is made up of amino acids. समझ आ रही है बात?
Yes, miss.
ठीक है?
Miss, and once it is complete, like the polypeptide chain is complete, does the mRNA fall off?
It falls off and it's destroyed.
Okay, miss.
mRNA also has a very short life. And okay, fine. I want to complete this. Who tells the cell that the polypeptide chain has now finished? Look, यहाँ पे देखो आप. अब ये रहा आपका राइबोसोम, ठीक है? mRNA is sort of you can see that the mRNA is sort of sliding, है ना? यहाँ पे ये ऐसा था. Look at the difference. यहाँ पे ये इतना सारा इस तरफ चला गया और यहाँ पे इतना सारा इस तरफ चला गया, which means that finally this should stop over here and the body will get rid and destroy the mRNA once the polypeptide chain synthesis is complete. Acha, now who will tell the cell that the polypeptide chain synthesis is complete? Just as we had a start codon, क्या था हमारा नाम start codon का? AUG. So I would say AUG is my start codon on mRNA. Similarly, we have stop codons also. Stop codons. And what are the stop codons? UR, UGA, and UAG. So, see, is this a stop codon? Yes, it is, because ये क्या है? UGA है. So, whenever the mRNA shows any of these three codons, UR, UGA, and UAG, it means the polypeptide chain synthesis will stop. खत्म. ठीक है?
Yes,
तो
miss.
बड़े मजे की बात है कि our DNA is such an amazing molecule that our DNA and RNA both are punctuated. Aren't they punctuated? It's starting with a letter and then it has a full stop also, and the full stop is what? You are, you ga, and you are. And the starting capital letter is AUG. That's the start codon. Right?
Yes, miss.
Okay. Achha, I think, let me just see. I think the only thing that is left from this chapter is mutations. And what else? Let me see. Mutations hai or that is it, and we'll be done with this chapter. So inshallah, achha, if I send you one more recording,